So I grew up in a small town in Eastern Europe, and in that small town, the circus will come once a year, and there was this ride that I loved, very simple ride. It will go up and down and around and faster. Now our very modest local playground had just some basic children needs, a seesaw, some swings, and a merry-go-round. So I thought, why not have the up and down and around all year long? So I convinced 2 strong boys to pull out the seesaw and put it onto the merry-go-round, and then we went off testing it. Great fun. It's just that due to the laws of physics, the 2 came together and snapped my finger. I looked at it not feeling any pain, but seeing the scared children in front of me, and then the pain hit me, and I splashed all of them, scaring all the moms in the neighborhood that afternoon. When I got home to my parents and told the story, my dad, otherwise a very collected man, said, "Ce a fost în capul tău?" which means, what was in your head? Well, now I took that question very, very seriously. Yes, and I've learned that the human brain has 100 billion neurons that are connected in 100 trillion ways. And that not all neurons are the same. No. There's many types of them. And even in a very small brain structure, such as the hypothalamus, there's dozens of different types of neurons that are involved in very different functions. And I don't know, but I think if you put an electrode there, you might stimulate all of those and the underlying behaviors, and I might end up very confused, with a very confused brain and body. So what I want to tell you about today is a technology that could allow us to stimulate just the cell types that we want and avoid everybody else. And this technology is based on light. You see, light is extremely versatile. Light is used to grow biological systems, as we know them. We use light to see biological systems, and you've seen the beautiful rainbow in the morning. And today I want to focus on the ability of light to control neuronal systems. And the way we do that is very interesting. So unlike electrical stimulation that indiscriminately affects all the neuronal circuits in the vicinity, when we use light on neurons, we can control only the neurons that are modified to respond to light. And how do we modify them to respond to light? We insert some proteins that are light responsive. And these proteins, they absorb photons of different colors. And they convert this energy into an ion flow. And this ion flow can either inhibit neurons or excite neurons. And we call these proteins OPSINS. They have rather remarkable origins, very humble. They come from unicellular organisms, from freshwater green algae, or from bacteria in salty lakes. And we take these proteins out of their modest origins, and we try to put them in mammalian systems. And we hope that they will function just fine. Now, we are asking a lot from these proteins. We're asking that they're going to be made in the right amount. We're asking that they're going to make it to the cellular membrane where they function, and we're asking that they still can control neuronal function. And this is not that straightforward. That's why it's rather remarkable that we are able to put these OPSINs into living brains from birth. So these are mice brains that had the opsin called channelrhodopsin for the activation of the neuronal activity throughout the brain, as you can see highlighted in green. So these mice are born with a foreign protein in them, and they're fine. So we wanted to push our luck even further, and we thought, oh, what if we put a fiber optic in the brain and deliver light to the brain and try to control these neurons in a very specific way? So we put the fiber optic on areas of the brain that are involved in motor control. And this is the result of the experiment. Light will turn off. You'll see a blue spot. Running around happily, just like me a while ago. And now the light turns off, and the rodent returns to its normal behavior, although it looks a bit confused, I would say. I would be as well. So while channelrhodopsin was very powerful to control the motor behavior in this rodent, we were very challenged by our inhibitory protein, halorhodopsin. Because of its origins, halorhodopsin, the protein that we use for neuronal inhibition, comes from a bacteria that lives in a very salty lake in the Sahara Desert. And the rules for survival are quite different from what we're used to in mammalian systems. So when we first expressed these proteins into neurons, they would aggregate and poison the neurons, as you can see in green. And it turned out that the proteins would be trapped in the endoplasmic reticulum, shown in red. And the endoplasmic reticulum is the protein factory of the cell, the mammalian cell. Bacteria don't have endoplasmic reticulum, so a protein from a bacteria wouldn't know how to get out of the endoplasmic reticulum and make it to the cellular membrane where it's supposed to function. So we had to help it a bit. We provided codes that would allow the protein to be exported out of the endoplasmic reticulum and into the cellular membrane so it can control neuronal activity. In a way, we provided them the zip codes to do that. So the result was beautifully expressed membrane protein, and that could trigger, just by using light, a flux of ions that would inhibit neuronal activity for very long time scales, and importantly, with red light. Red light is very good. Just think about ultraviolet that damages your skin. Red light is much safer. And in addition to being much safer, also penetrates the tissue much better, which allows you to control larger brain areas by the use of red light. Although, in reality, we would like to have OPSINs for all different colors, because as you've learned today, there's many different circuits that do amazing things, so we'd like to have the ability to use different colored OPSINs to control different circuits at the same time. And the way we can find new OPSINs is either to look in nature and explore the bounty of nature and look for natural sources for these proteins, or engineer them, force the proteins to respond to new wavelengths and have other interesting properties. So what did we do so far with these proteins? We did experiments trying to reveal some of the mechanisms of the brain stimulation. For example, we were able, by modifying motor circuits in rodent, to restore motor symptoms in a Parkinsonian model. And the way we did that was to introduce OPSINs by using viruses. Viruses are nasty, they infect us, we don't like them very much. But in our lab, there are allies, because we can package the OPSIN DNA into a virus and inject it to a very local brain area. And in front of the OPSIN DNA, we can put a short sequence called promoter. And this promoter decides what cells are going to make the OPSIN and what cells are not going to make the OPSIN. So by using these methods, we can effectively genetically extract brain areas that otherwise would look just like their neighbors. So when we put the light into living brains, we can control only those specific circuits. And the result of some of these studies pointed to the fact that deep brain stimulation, in addition to controlling cell bodies at the stimulation side, it's very likely to also affect fibers of passage, axonal cables that go into that region and out of it. And for example, the subthalamic nucleus, the preferred nucleus for deep brain stimulation, receives many fibers from cortical areas. So just a thought, if that's the case, can we place the electrode more superficially, and in this case, maybe bypassing some of the challenges associated with inserting things deep within the brain? And the other question is, what if not? What if subthalamic nucleus is such a good target for deep brain stimulation? Just because it receives all of these fibers, it's a convergent point for all of these. So we can start to answer some of these questions by using optogenetics. And I want to remind you of the origin of these modest tools. They come from unicellular organisms, and they're very, very powerful. And they have names like Chlamydomonas reinhardti, Natronomonas pharaonis. It's not their fault, people named them. But we discover more and more, and we hope that we're going to expand our tools so we can start to understand the brain better and better. And I would like to acknowledge the hundreds of people that worked on growing optogenetics over many years and using it to understand some of the mysteries of the brain. And also leave you with a confession. I still don't know exactly what's in my brain, sorry, Dad, but I'll keep researching. Thank you for listening.